Right now, we do see your presentation mode, not the full screen presentation.
Okay, let's see. Let me end for a second? And we will share the other screen. How about this one?
All good.
All good. Wonderful. Okay, good morning or good evening, depending on where you are. I'm very excited to have this opportunity to share some of the work that our lab is doing. I'm just going to try to move this hide floating meeting controls. Wonderful. Okay, now I can see what I'm doing. All right, so I was actually hoping to start with an observation that I think may be relatively banal for this group, which is the observation that microglia are found everywhere in the central nervous system. So this is just a visual representation of that. This is a coronal brain section from a young adult mouse that I think really helps visually highlight that these cells are found in every part of the central nervous system. And what this implies is that any population of neurons in the brain is going to be surrounded by and in close proximity to microglia. So to illustrate that a bit further, I want to show you one group of neurons that is one of my favorite groups of neurons. These are dopamine neurons that are found in the ventral tegmental area and the substantia nigra pars compacta. These neurons are very important for learning about reward or learning about aversive stimuli. They're important for cognitive flexibility and also, of course, movement. I think they're very beautiful just in and of themselves, but we know that they're not by themselves in the brain. They are completely surrounded by and permeated by microglia and also, of course, other glial cells. So this, I hope, also just reinforces this idea that probably is well accepted to all of us on the webinar that glial cells, including microglia, are really creating the environment that all neurons live and operate within. So given that, how can microglia be potentially influencing neuronal function and health? Again, this may be somewhat repetitive or a unnecessary introduction for this crowd, so we'll go through it quickly. At this point, we know that there are many ways that microglia can impact tissue health through their constant surveillance of the surrounding tissue and removal of pathogens or debris or toxic proteins. And we also know that microglia have many mechanisms for interacting with synapses, affecting the number of synapses or receptor composition at those synapses, as well as mechanisms for influencing neuronal excitability. We also know that they can influence overall neuron health through secreted factors. So that could be trophic factors during health, but also in the context of any insult to the CNS. Microglia are rapid responders. They will release pro and anti-inflammatory signals as part of these responses. As part of responses to insults, they can also encapsulate areas of damage or interact with disease-causing aggregates and play important roles in signaling to other glial cells and immune cells to coordinate responses to any CNS pathology. So I think taking all these abilities of microglia together, you can really make a strong argument that microglia play a critical role as kind of setting the backdrop for neuronal functioning and their resilience and vulnerability throughout the lifespan. And this is probably an idea that many of us have been on board with and accepted for many years. But at this point, I think most of the neuroscience community is also on board with this idea that microglia are influencing neuronal health and function in important ways. So I want to circle back if we're, all on board with the idea that microglia impact neuron health in important ways. Let's circle back to the idea that they are found all over the central nervous system, that they're interacting with neurons all over the central nervous system, and add a layer of nuance here, which is that even though microglia are found throughout the central nervous system, they're not identical across brain regions and CNS regions. So we and a number of others have shown that across brain regions, there are important differences in microglial cell density, as well as their branching complexity. There are important differences in their patterns of gene expression, that there are differences in their cell process motility, and also phagocytic capacity, among other cell features. So this really, I think, raises the possibility that the specific ways that microglia are interacting with neurons and influencing their health and function is potentially region specific and varying across brain regions. And so we really need to dig in and understand how microglia are affecting neuronal health and function for each neuron population that's out there. I want to show you a little bit more about what these regional differences in microglia look like within the basal ganglia circuits, because these are the main circuits where my lab has spent a lot of time studying microglia and the way that they may impact neurons. And these are circuits where these regional differences in microglia are really quite evident. So what you're looking at here are brain sections from a 2-month-old healthy mouse, the CX3CR1 GFP mice. And we're looking at 4 different nuclei of the basal ganglia circuits. So this is nucleus accumbens or ventral striatum. It's an input nucleus for these circuits. We've got the ventral tegmental area and the substantia nigra pars compacta. Those are the regions where the dopamine neurons live. And then we have substantia nigra pars reticulata, which is one of the output nuclei for these circuits. And I hope that even just looking at these images and not even looking at the quantification, you can appreciate that there are pretty pronounced differences in how abundant microglia are across these nuclei. There are also differences in how complex their branches are. So there's a much more complex branching in the nucleus accumbens and the reticulata, a more sparse branching here in the VTA and the SNC. We can even see regional differences in the abundance of lysosomes within these cells. Again, this is just a young adult, healthy mouse. What we find is that there are some regions like the SNR where microglia have a lot more lysosomes relative to their cell volume. And then here in the VTA and the SNC, they have fewer lysosomes relative to cell volume. We've also shown that there are important regional differences in transcriptome of microglia across these nuclei. And so this is just meant to give you a little bit more of a sense of what I'm talking about when I say that there are regional differences in microglial properties, even in young adult healthy mice. So what might that mean for microglial interactions with nearby neurons? That's something that my lab is very interested in, and we've spent a lot of time and individual lines of research trying to dig into. What I'm hoping to tell you about today are 2 lines of research going on in the lab, one that takes place during CNS development, specifically postnatal development. And I'll tell you about some findings we have where we think that in particular, the microglia in the nucleus accumbens may be equipped to promote synaptogenesis in this brain region. And that ends up having consequences for anxiety-like behaviors. And then at the other end of the lifespan, and we'll try to keep an eye on time. I know we got started a little bit late. I'll tell you about some work that we've been doing looking at how microglia change during aging. And in particular, there are some unique things that we're seeing in the VTA and the SNC microglia that we believe may be relevant for aspects of cognitive decline during aging. So let's start with what microglia are doing in the nucleus accumbens during development. hopefully most folks are familiar, but just in case, I'll make sure that we all know that microglia do have distinct developmental origins from the other major glial cells within the central nervous system. So astrocytes and oligodendrocytes are arising from the same progenitors that are giving rise to neurons, and they're coming about within the developing neuroectoderm. The microglia instead are coming from these primitive macrophage progenitors that originated in the yolk sac. And after invading the developing nervous system in early embryonic periods, they need to then spread out and colonize the brain with its population of mature microglia. So, given that microglia invade the nervous system and then need to migrate and proliferate, we were interested in when is it during development that these regional differences that we had observed in basal ganglia circuits, when is it that those come about? And Keenan Hope, a former technician in the lab, decided to look at this with a particular focus on the ventral tegmental area and the nucleus accumbens. And he started just by looking at microglial density at a number of different postnatal periods and then out to 2-month-old mice. And he ended up finding something that we weren't necessarily expecting, which was that it seems like microglia are overproduced in both of these brain regions. So they'll have sort of peak density around the second and third postnatal weeks before dropping down to the density that we observe in young adult animals. And he also observed that this overproduction in the nucleus accumbens was particularly pronounced. So, in fact, if we normalize everything to the density that's ultimately achieved in young adulthood, you do see that this density, this increase in density and overproduction is greater in the nucleus accumbens than it is in the VTA. We were able to show that this increase in density in the nucleus accumbens here between P8 and P12 is due to a lot of microglial proliferation. And then this decrease in density on the other end is due to a combination of ongoing brain growth, but also programmed cell death of the microglia. And that was useful in sort of telling us what was shaping this abundance of microglia during this period, but it didn't really tell us why would it be that there are so many microglia during this second and third postnatal week. You know, it's possible that they just sort of overshot the number of microglia that was needed and then that got corrected. But we were curious whether there was actually some purpose for this elevated number of microglia in the nucleus accumbens during this period. So to look at that a bit further, we made use of mice. Morgan, I might also ask you if you notice that people are unmuted. Maybe you can mute them too. I think you are able to do that as a host. So to look at this a little bit further, we were interested in whether or not there's a relationship between this period when there are a lot of microglia and synaptogenesis, because this is a known period of synaptogenesis in this brain region. To look at this a little bit further, Eric Mocha, who is also a former lab assistant, decided to take an electrophysiological approach. And he utilized the fire mice, which were generated by Claire Pradans, which many of you are likely familiar with. These mice lack microglia throughout their life. And so what Eric did is he prepared acute brain sections from these mice as well as their littermate controls and did whole cell recordings from medium spiny neurons. Those are the main neurons in the nucleus accumbens. And he looked at miniature excitatory postsynaptic currents, miniature EPSCs. And so each of these downward deflections represents just a spontaneous synaptic signaling event. And you can generally take the abundance of these events or the frequency of these events as an indicator of how many synapses the neuron is receiving. So these are some examples of these recordings from medium spiny neurons in control mice. when Eric recorded from mice that lack microglia, we could see there are definitely still synapses forming. But when you quantify what's going on here, what you can see is that this miniature EPSC frequency is kind of steadily marching up in control animals, whereas in the mice that are lacking microglia, there really are a lot fewer of these miniature EPSCs. Another way of looking at that is with a cumulative probability distribution. And so particularly at this P14 and P20 period, what you can see is that there's a longer inter-event interval between synaptic events in these knockout mice. So if microglia aren't there, we basically seem to be having fewer of these synaptic events. Things are largely back to normal out here at 2 to 3 months, at least in terms of the number of synaptic events that we're observing. We've done a number of things to dig into this further and just see what's going on there. So for example, we looked at presynaptic function to see how that might be affecting the number of synaptic currents that we see. And we don't see any major differences in presynaptic release probability, except at P22, where we actually saw a reduced paired pulse ratio, which means an increased probability of release. So we think actually, if anything, this lower number of synaptic events that we're seeing, the system might be trying to compensate by increasing presynaptic release probability somewhat. We did also look at inhibitory synapses, and we're not seeing major differences there in terms of the frequency of inhibitory synaptic events. So it seemed like the absence of microglia in the nucleus accumbens was primarily affecting excitatory synapses and resulting in a reduced number of those synapses. To confirm that, we tried to take an independent approach, so just using immunostaining for presynaptic and postsynaptic compartments, focusing on the excitatory synapses and this period where we had really observed the biggest decrease in miniature EPSCs. And we're finding similar things using histology. So if you just look at the abundance of the presynaptic puncta or the postsynaptic puncta, that was reduced in the mice that are lacking microglia. And also if we just focus in on instances where we've got co-localized pre and postsynaptic puncta, that's also reduced in the knockouts. So I told you that it seemed like the synapse number was back to normal, more or less, by the time you got out to 2 to 3 month old mice. Does that mean that this is really just something that impacts development and everything's kind of normal by the time we get out to adulthood? We don't necessarily believe so. We've done some behavioral analyses with adult mice and find anxiety-like phenotypes using either the open field test or the elevated zero maze test. So the mice that lack microglia are spending less time in these open areas. The nucleus accumbens is a brain region that is important for anxiety-like behaviors, but of course there are also other brain regions involved. We've also looked at this more elaborate modified fear conditioning behavior that the DiNardo lab uses extensively. So this is a collaborative project with the lab of my colleague, Dr. Laura DiNardo, who's also here at UCLA. So in this behavior, the mice are in this chamber. They receive 9 pairings of a tone and a foot shock. And they learn that they can avoid this foot shock by getting up on this little safety platform that's here in the corner. We can then test the behavior of the mice 1 day later. You put them back in this chamber and only play the tone and see whether they remember that they should get up on the platform to avoid this this unpleasant shock. This is a little bit of, it provides you with a lot of nice opportunities to analyze behavior beyond standard fear conditioning, where you generally just have to look at, are the mice freezing? Here we see, have they learned this action that they need to take in order to avoid the stimulus? So during the training day, the mice that lack microglia, they perform pretty similarly to the mice that have microglia. So if we look at how much time they spend on this little safety platform, or how many of the specific trials they successfully get up on the platform to avoid the shock, you can see that as we go through more and more pairings of this tone with the shock, the mice are learning they've got to get up on that platform. And if anything, it looks like the mice that are the fire mice are potentially learning this more effectively than the mice that have microglia. But the next day, when we ask if they remember this, the knockout mice are not performing as well as the controls. So they're spending less time on the platform when the tone is played compared to the control mice. We can also see that they're taking a longer amount of time after the tone is played before they're getting onto the platform. They don't show any differences in just the amount of time that they're spending freezing. So we don't think that there's necessarily any difference in this sort of innate fear, but this learned avoidance behavior is not being effectively brought about in the knockout mice. I told you that the nucleus accumbens is important for these types of anxiety and fear learning behaviors, but that it's not the only brain region involved in those behaviors. So we did want to also do some additional experiments to see whether it was truly potentially changes in the nucleus accumbens that are involved in some of what we're seeing here. So we did some fiber photometry where we put a calcium indicator in the medium spiny neurons in the nucleus accumbens. So we can look at activity of those neurons during the behavior. And if you remember, the mice that lack microglia were learning that avoidance behavior a bit faster. And indeed, on the training periods, we see that the mice are, if anything, having a larger calcium increase, a larger increase in activity of the medium spiny neurons at the time that they receive a shock. So here you can just see this quantified as the area under the curve. So that's consistent with what we were seeing behaviorally. But then back on this day when they were having difficulty remembering to get up on the platform, the retrieval day, we see that the wild type mice, when they go onto the platform, are having a clear increase in calcium signals, whereas the mice that lack microglia really pretty much aren't showing anything. So this also is consistent with what we were seeing behaviorally, that the knockout mice on the retrieval day are not really remembering that they should get up on the platform. We can also see this effect just by using FOSS staining. So on that retrieval day, if you play the tone, the mouse is supposed to remember that the tone means a shock and get up on the platform. And then we sacrifice the mice 90 minutes after that retrieval and do immunostaining for FOSS in the nucleus accumbens. We can see that there are fewer FOSS positive cells in the nucleus accumbens in the knockout mice. So we're not saying that the nucleus accumbens is the only brain region involved here, but certainly there are lasting changes in activity that's associated with behaviors in the nucleus accumbens in these mice that lack microglia. The last thing we did here is an experiment that it's not my favorite experiment, but it's, you know, we in the microglial field, we're always working with the tools that we have at our disposal. We also tried to carry out some transient depletions of microglia that are also region specific. So we used injections of clodronate disodium salt. So we injected this bilaterally into the nucleus accumbens at P10. And what you end up seeing here is that at P15, so 5 days after this injection of clodronate, we can find these regions of the nucleus accumbens that are devoid of microglia. You can also see this edge of microglia near that empty region that are getting ready to repopulate it. And in fact, by P21, can you mute your phones, please? Thank you. By P21, the microglia have repopulated and, are back to more or less normal density. So in these mice with a transient depletion, we did still see some of these anxiety-like phenotypes in adult mice. So that includes spending less time in the open portion of an elevated zero maze. We still saw this apparently like enhanced learning to get up on the platform during training for that fear avoidance. But we weren't necessarily seeing that same deficit in retrieving that learned avoidance. So no differences here in when we're trying to play the tone and just see if the mice remember to get up on the platform. So this could indicate a number of different things. It could mean that we would need to deplete microglia during a slightly different time window in order to see this impact on the retrieval of this learned avoidance. It could also indicate that the execution or retrieval of this memory it involves other brain regions besides just the nucleus accumbens. Or it may mean that this particular effect is not necessarily driven by what's going on with microglia. So I want to summarize this first, what I've told you so far is that within the basal ganglia circuitry or the reward circuitry, microglia are overproduced during early postnatal development, and that was especially true in the nucleus accumbens, which led us to investigate microglia in that brain region a bit further. This period of microglial overproduction in the NAC is aligned with a period of synaptogenesis in that brain region, and we found that synaptogenesis was impaired in mice that lack microglia. And it does seem that the absence of nucleus accumbens microglia during development is associated with anxiety-like behaviors. So this was intriguing to us, and where we want to go, where we've been going next with this is trying to understand whether the impact of lacking microglia on the developing nucleus accumbens will be similar to the impact of some sort of early life insult that perhaps alters microglial function or perturbs microglial ability to support the maturation of these circuits. So still in collaboration with the DiNardo lab, we've been looking at how a model of early life stress impacts microglia within the nucleus accumbens and associated behaviors from these that depend on these circuits. We're also, my graduate student, Megan, is particularly interested in astrocyte microglial dialogue. And we have some reasons for thinking that glial-glial dialogue may play an important role in regulating microglial development in this brain region. And so she's been looking at both how this dialogue may be important for normal microglial maturation, but also whether this is something that's perturbed by early life stress. So this is work that's in progress, but I still want to just give you some early indicators of what we're seeing. This is the model of early life stress that we're utilizing. It's called limited bedding and nesting for a relatively brief period from P4 to P11. There's a change in the availability of bedding nestlets, those cotton nestlets within the cage, and there's also a wire mesh on the bottom of the cage. What this ends up doing is it causes the dam, the mother, to be more active in her movements. She's kind of going around and trying to search for nesting material. So there are a lot more transitions on and off the pups. This does result in lasting behavioral changes in the pups. So when these pups are allowed to grow up and get weaned, they have anxiety-like behaviors seen here in open field, as well as other behavioral changes. So there are a number of behavioral changes after this early life insult that are similar to what we saw in the mice that lacked microglia. In terms of what's happening to microglia in the nucleus accumbens in this model of early life stress, there are some subtle changes in microglial density. So maybe a little bit of an increase in microglial density in these mice that had the limited bedding and nesting at P22. And perhaps this is more accentuated in the nucleus accumbens than other brain regions because we weren't really seeing this kind of thing in the prefrontal cortex. We can also see some very subtle changes in microglial morphology, again, primarily at P22 and P40, where actually the microglia from the early life stress mice were a little bit more complex. So they're showing a little bit more branching complexity. But these are relatively, these are relatively subtle changes in morphology and density. Does that mean that the microglia weren't necessarily that impacted by the early life stress? Absolutely not. We have also looked at the transcriptome of the cells. So we've isolated nucleus accumbens microglia from mice that either had normal rearing environment or the limited bedding and nesting at a number of different developmental time points and carried out bulk sequencing. One of the things that I want to highlight here is out here at the adult time point. If you're comparing nucleus accumbens microglia from mice that had this normal rearing or the limited bedding and nesting rearing, we find a surprisingly large number of differentially expressed genes out at P70. So the animals have grown up. Microglial density and microglial morphology is normal, but that does not mean that their transcriptome does not have a lasting consequence of this early life experience. In this group of mice, we have also collected astrocytes. There are changes in astrocytes as well, but these are not as extensive, at least in terms of the number of differentially expressed genes. And we're very interested, again, in looking at signaling relationships between the cells. I'll just finish this portion by highlighting some of what we were finding in terms of the specific genes that were impacted by early life stress in the microglia. Both at P8 and at P70, we were seeing changes in expression level of a number of genes that are implicated as being major regulators of sort of overall microglial phenotype. We see genes that are involved in phagocytic function. And then in terms of genes that could be involved in microglial sensing of neuronal activity or mediating cell-cell contact, that maybe seemed to be a little bit more prominently impacted at P8 during the stress. And then we were seeing a lot of microglial genes that are implicated in regulating the extracellular matrix or potentially remodeling the extracellular matrix. That was more prominent at P70. And if we just do pathway analysis of all of the genes that were impacted by the stress at P8 or at P70, a number of interesting things are showing up here, including phagocytosis, regulation of synapse structure, regulation of cell matrix adhesion, et cetera. This is a data set that we're still very actively analyzing and digging into, but I was just hoping to give some early flavors of what we're seeing. And I think even this early analysis supports the idea that early life stress definitely has a pretty pronounced impact on microglia in the nucleus accumbens, and it's impacting them in ways that we would predict will affect microglial support of synaptic plasticity and likely may be involved in how these mice are behaving, how these circuits are functioning, and how the mice are behaving in adulthood. We're also very interested in if the microglia have this really profound, lasting change after early life stress, particularly in the nucleus accumbens, what does this mean for how they may respond to second hits later in life, whether that's additional stressors that they might experience in adulthood or adolescence or drug exposures, for example? Is there an important way in which microglia may be acting as a link between individuals who experience early life stress are often at higher risk for various psychiatric conditions, including addiction and problems with anxiety. So is there an important role for microglia in mediating part of this link is what we're hoping to continue studying. And as I mentioned, we're actively looking at glial-glial dialogue in this context as well. So that's what I was hoping to tell you about nucleus accumbens microglia during CNS development. I think we're doing okay for time, especially if people are willing to stay like 5 minutes over. And so with that, I can switch gears and tell you a bit about what we've been finding in terms of microglia at the other end of the lifespan during aging. At the other end of the lifespan, we were also initially starting to look at microglia during aging simply to understand if those regional differences in microglia that we saw in young adult mice persist throughout the lifespan. You know, this lower density of microglia in the VTA and higher density of microglia in the nucleus accumbens, is that just there in young adults, or is that something that's there lifelong? And so Eric and Keenan were working together to look, again, just starting by looking at microglial density across a relatively wide portion of the mouse lifespan. Out here at 22 months, this is probably like an 80-year-old human, 18 months, more like a 65-year-old human. And they were comparing what they found in nucleus accumbens to what was found in VTA and also SNC. So nucleus accumbens, there's definitely an increase in mouse-to-mouse variability, but no major changes in microglial density. In the VTA, it's a different story. They start out with lower density of microglia, but beginning in midlife, 13 months, you see this progressive increase in microglial density. And very similar things were happening in the substantia nigra pars compacta, that other portion of the brain where dopamine neurons are found. We were interested in what might be driving this proliferation of microglia within the VTA and the SNC. And one of the main things that we've ended up following up on is what's happening with the microglial lysosomes. This is an example of what lysosomes look like in microglia in a young adult mouse. You know, you see these CD68 positive puncta just scattered all throughout the soma and the cell processes. As many of you are probably familiar, microglial lysosomes change a lot during aging. So here's, for comparison, from an 18-month-old mouse, you see these much more swollen, large lysosomes in the cell soma and a loss of these lysosomes that are out in the cell processes. So we were intrigued when we looked at this and found that these changes in lysosomes are more severe in the VTA, where the microglia are proliferating, compared to the nucleus accumbens, where they're not. So, for example, in the VTA, this average volume of individual lysosomes is larger. They have lost more of their cell process lysosomes than the microglia in the nucleus accumbens. Along with these changes in the lysosomes, we were observing autofluorescent aggregates that were filling up the microglial lysosomes. So many of you have probably also heard of these autofluorescent aggregates as well. They're also referred to as lipofuscin or aging pigment. This is material that accumulates in neurons and also microglia during aging. And the aggregates in the microglia can really be quite pronounced and angry looking. If we also just look at the volume of these aggregates, which are primarily found within the microglial lysosomes, we see kind of the same thing, that these aggregates are bigger in the VTA microglia than they are in the nucleus accumbens microglia. And of note, the aggregates that are in the microglia are much bigger than the aggregates that you see in other cells, such as neurons. So this material, lipofuscin, generally has been viewed as a benign phenomenon that just emerges during aging. These are thought to be damaged lipids and proteins that normally would get degraded by the proteasome, but then during aging, that breakdown starts to be less effective, and you eventually get these aggregates of damaged lipids and proteins that are trafficked to the lysosome. But it was largely viewed as something that's just kind of a benign thing that doesn't really affect the cells. We think that that definitely needs rethinking, particularly in the context of microglia. In the VTA, we think that there are important links between these aggregates and the fact that the cells are starting to proliferate. So if we look at proliferating cells in the VTA and SNC of the aging mice, what we can see is that usually when the cells divide, these autofluorescent aggregates go to one daughter cell, and then the other daughter cell is left relatively aggregate free. And you can see this here. This is just sort of the volume of the aggregate, how much of it ended up in each daughter cell. Occasionally, you'll see these examples of the aggregate being split relatively evenly, but most of the time you have a situation where there's one daughter cell that has a lot of aggregate volume and the other one that has almost nothing. So this could be an adaptive response where the cells are dividing and trying to dilute out this material. What are additional reasons why we think this accumulation of lipofuscin in microglia is important and should be further studied in the context of aging? One reason is that we've also found that the accumulation of these aggregates has a really pronounced impact on the microglial transcriptome in a region-specific manner. So one nice thing about these aggregates is that you can see them by histology, just by leaving one channel empty, but you can also see them by flow cytometry. So if you are doing flow cytometry and you gate your microglia and then you just look at their autofluorescence, here we've got red and far red autofluorescence. And you do this even from middle-aged mice. So we've done this from 13-month-old mice. You can find microglia that have high autofluorescence as well as microglia that have low autofluorescence. And so we used this to separately collect the microglia that have high and low autofluorescence from two different brain regions of 13-month-old mice. And then we did bulk RNA sequencing to look at their transcriptome. What you can see here is that the microglia that have low aggregate, we can appreciate some regional differences in their transcriptome. But what really surprised us was how different the microglia that have high aggregates are across these brain regions. I might have predicted that there are regional differences in microglial transcriptome, but once they have to start dealing with this aggregate burden, maybe they kind of shift into a similar aggregate loaded state. That is not what we were finding. The microglia in the midbrain, where the VTA and the SNC are, that have high aggregates are very different than the microglia in the striatum that have high aggregates. Just to give you a little flavor of what the differentially expressed genes in this data set contain, there were perhaps unsurprisingly a lot of genes that are involved in lysosome function that are differentially expressed at different levels between microglia that have high and low lipofuscin. So that reinforces this idea that these aggregates are really impacting microglial lysosome state. What was interesting to us also was that the way that these aggregates were changing expression of lysosome function genes was not the same across the brain regions. So one of the major examples of this would be cathepsin K. It was majorly upregulated in midbrain microglia that have high aggregate and really downregulated in striatum microglia that have high aggregate. So the way that microglia responded to these aggregates is really different across the brain regions. We also see some interesting things if you look at pathway analyses. So these would be differentially expressed genes that were shared in midbrain and striatum. So similar things are happening with the microglia that have high aggregate in both of these brain regions. And we see lysosome showing up there, endocytosis and phagosome to us are suggesting that the accumulation of this material is likely not purely driven by what's happening inside the microglia, but also informed by their uptake of materials from the extracellular space. Interestingly, in the differentially expressed genes that were unique to the midbrain microglia, so we're really only seeing those in the midbrain microglia with high aggregate, we see mitotic cell cycle. Maybe I had some arrows here. Sorry, I had some arrows and I'm forgetting to click the animations. We see mitotic cell cycle and DNA replication checkpoints. So that's consistent with this was the brain region where the cells were really proliferating the most as a result of this aggregate burden. And then it was certainly of interest to us that, there are a couple pathways showing up here that are related to synapse status and synapse activity. So hints that these aggregates may be changing microglia in a way that's relevant for synapse function. Okay, and so then to quickly poke around a little bit at, is accumulation of this material relevant for synapse function? We've gone about that more from a behavioral angle. So can we see at least whether there are links between this material and cognitive status in aging mice? One way that we've done that is using a behavioral paradigm that project scientist Daniel Gray devised. So just briefly, mice are in an arena where there's a little well that has food treats and they can dig for the food treat. We take them out of the arena and then put them back in the arena after a short or long delay. And in this period, there's the original rewarded food well, but also 3 distractor wells that don't have any reward. And we will monitor the behavior of the mouse and see if it remembers where the reward was. The mice are doing this every day, and each day there's a different location that's rewarded. So they need to have adequate cognitive flexibility to say, like, okay, what's today's reward location? So because it's reward-based and involves cognitive flexibility, this task is for sure engaging the dopaminergic circuitry. We've done this task for using both young adult and late middle age mice. These are just C57 Black 6 wild type mice. If there's a short delay, the old mice are performing just as well as the young mice. But if there's a longer delay, the old mice are not performing as well. So they're making more errors before they go to the correct well. You can also, for an individual mouse, look at the difference between how well it did with the long delay versus the short delay. That's this difference measure. That really highlights how the older mice are not doing as well. We can also look at this in other ways. So the older mice are spending less time at the proportion of time at the correct well is reduced compared to time that they're spending at other wells. One other thing I want to highlight here is that there is a lot of mouse-to-mouse variation here. So we can see mice, older mice, that really are performing a lot worse than the younger guys. But there are also older mice who are performing just as well as the younger mice. And so this gives us the opportunity to go in and analyze tissue from these mice and ask, what's going on with microglia and microglial lipofuscin in the mice that are still doing relatively well compared to the mice that are having cognitive decline. One of the things that was very easy to look at was microglial density. Again, we think that these aggregates, the lipofuscin aggregates, are pushing the microglia to proliferate, particularly in the VTA. So in these cohorts of mice, we can see that same thing, that microglial density in the VTA has increased with aging. And now if we look at how this relates to behavior, what we're finding is that even just looking at the 18-month-old mice, the mice that had more microglia are doing worse on the behavior. So something about this proliferation and what's going on with these aggregates appears to be associated with worse performance in this behavioral paradigm. Something else that we've done, trying to more directly link the lipofuscin itself to cognition, not just proliferation that we think is driven by lipofuscin, involves two different proteomic approaches. So bear with me. The first thing we did was we wanted to know what are the proteins that are present in those aggregates. So we devised a way to purify microglia, lyse them, and then from the lysate, purify out those autofluorescent aggregates. This is what they look like when they're cell-free aggregates. And we sent that for proteomics, and we were able to identify a little over 400 proteins that are part of these microglial lipofuscin aggregates. So now we know what the proteins are that are in those aggregates. Can we use that to look at the status of lipofuscin in mice that we have information about their cognitive function? For this set of experiments, we did a bit more standard behavioral battery. So we had mice running through open field, novel object recognition, and T-maze, which also tests cognitive flexibility. And then we use the behavioral readouts from all of these tests and put them into just an unbiased clustering to ask which mice are overall behaving most similarly to one another. We can see that most of our young mice that we ran through this paradigm cluster together. And then the older mice, there's a chunk of them that cluster with the young guys. And there's a chunk of them that are clustering separately from the young guys. And we're referring to these as unimpaired or impaired 18-month-old mice. We collected microglia from those behaviorally phenotyped mice, particularly from the midbrain, again, where the VTA and the SNC are. We did this using the CD11B magnetic sorting, and then did proteomics of the microglia from these mice. Now, can we ask, what are the lipofuscin proteins doing in these microglia in mice that have impaired cognition or unimpaired cognition? And this is just an early look at what we're finding. There were, of those proteins that we identified that are major components of lipofuscin, 104 of them had a significantly different level of abundance between the impaired and unimpaired mice. Most of these were more abundant in the impaired mice compared to the unimpaired mice. There were a subset of these proteins that were more abundant in the unimpaired mice, but the majority were more abundant in the impaired mice. And I think this is compelling evidence that the status of these lipofuscin aggregates in microglia is not totally benign. If you have more of these aggregate proteins in microglia in your midbrain, that may be impacting the rate of cognitive decline during normative aging. So we're excited to continue following up on these findings, and I'll just quickly summarize what I've told you about this aging portion of the talk. We showed that microglia in the VTA and the SNC begin proliferating during aging, and that's something that happens in midlife. It's not just way out at the end of life. Something that appears to potentially be driving that proliferation is the accumulation of these autofluorescent aggregates in microglial lysosomes. That process is more severe in VTA microglia. These aggregates are associated with really pronounced changes in microglial transcriptome. I didn't have time to tell you about this, but we've also used genetic approaches to just drive lipofuscin accumulation in microglia separately from aging. That's using some CLN3 knockout mice that we obtained from Andrea Balabio. And that is also sufficient to induce microglial proliferation and is more severe in the VTA. And finally, we believe that the accumulation of these aggregates in microglia in the midbrain is relevant for the status of these circuits. And we're also very interested in how these aggregates and microglial phenotypes that are associated with the aggregates intersect with risk for neurodegenerative disease, as aging is the biggest risk factor for most neurodegenerative diseases. So hopefully I was not talking too super quickly. And if anyone has to run, I totally get it because we started a little late and went over time. But thank you so much for your attention. I have to thank all the grad students and postdocs in the lab who drove this work, and our collaborators, in particular, Laura DiNardo's lab and Claire Perdan's. So thank you all for your attention, and I'm happy to take questions. I don't know how you all typically do it, if you put them in the chat or...
Oh, okay. Thank you, Lindsay. Actually, we have a lot of questions in the chat. I will read them for you, and then if someone wants to make the question themselves, like Leticia, she can speak. I will start with the first one. This one is from Linus Alshavsky. He said, did you observe any locomotor-related deficits upon wiping out microglia in the nucleus accumbens using clodronate? And how do you tie together the lack of changes in the basal neurotransmission in the adult nucleus accumbens with the behavioral changes observed?
Yeah, those are great questions. We did not specifically assess locomotor behavior. Either in the fire mice or in the context of the clodronate depletion. There are definitely ways to do that. It might be a little trickier in the early postnatal period when we're doing the like shortly after the clodronate deprivation. But certainly once the mice are reaching weaning age and adulthood, there are a lot of nuanced ways that we could look at that. We haven't done that. It's a nice idea. The fact that the number of synapses appears to be back to normal in adulthood, but that we're still seeing behavioral deficits. Our interpretation would be that even though the number of synapses is normal, the specific patterns of connectivity are very likely altered. So the nucleus accumbens receives both cortical and subcortical inputs and modulatory inputs. And we have not specifically looked at whether there are changes in the relative balance of inputs that are coming into the nucleus accumbens, but there are also great like viral tools for doing that. Honestly, I think we are primarily interested in continuing to look at this model of early life stress and understanding how that may perturb synaptic function, synaptic inputs, more than trying to fully flesh out what was happening in the fire mice.
Okay.
Thank you for that question.
Okay, the next one is from Alberto Gomez. He said, thank you very much for your great talk. Is the reduction in synaptogenesis directly due to the local absence of microglia in the nucleus accumbens? Or are there other brain regions projecting to the nucleus accumbens also undergoing similar changes that could contribute to or modulate this reduction? I think it's related to the previous question.
Yeah, those are great questions also. We have not extended our analyses to other brain regions in the fire mice. One thing that we have done, however, is that in fire mice and controls, we did do some tissue proteomics, at the P22 time point. So that was when we were really observing the largest decrease in miniature EPSCs. We wanted in particular to look at astrocyte-derived synaptogenic cues, as well as some of these like adhesion proteins that are really important for supporting synapse maturation. Because one possibility would just be that with microglia absent, something about these known synaptogenic cues may have been altered. We weren't finding any really strong indications that those known synaptogenic cues are perturbed. Some of the astrocyte synaptogenic cues were like below the sensitivity of our instrument. There were some astrocyte proteins in that data set that were altered, which maybe isn't surprising. So I think we can't completely rule out that something has been altered in astrocytes as a result of microglia being absent. But I think it also raises the possibility that microglia are directly supporting synaptogenesis in some way. And we did have a few protein candidates that we think might be worth looking into further. I'll just stop there for that question. Thank you for that question.
Okay, now I have several questions from Paul Lucasen. I will read them in order, just one by one. The first one is, thank you for a very, very clear talk. For the early life LBN model, most people use earlier time points, like P2, P9. Why did you choose your specific later time window? Is there any microglia specific reasons?
Yeah, thanks for that question as well. I think, as is often the case, the answers to some of these questions are largely pragmatic. So when my collaborator, Laura DiNardo, and I were embarking on wanting to delve into early life stress, we interacted a lot with Tali Barham, who's well known in this field, and we ended up going with the specific paradigm that her lab has used because they have such a large body of data about how it impacts different behaviors and different molecular players. I'm certainly aware that there are other variations of the stress paradigm, and honestly, it could be interesting for us in future directions to see how much any like microglial phenotypes that we're seeing are really dependent on the specific window during which the stress happened or not. I think that could be interesting to explore. But the main answer is that we were working with the advice and large prior data set of our colleague down the road, Tali Barham.
Okay, there's another question from him. Regarding your omic analysis and the multi-hit plans after early life events, any thoughts or speculations on what might represent the key, the set of genes, factors that prime young microglia to perform better later in life?
Yeah, this is not going to be a very satisfying answer, but I don't think we have any, I don't think we're far enough along in digging into the data set that we have to make really good speculations about, are there key transcription factors, or were there chromatin modifiers that have been impacted by the stress that would be relevant to think about in terms of like priming the microglia to act differently. So I don't think we have any concrete speculations yet, but there's a lot of, lovely work out there in the field by microglial colleagues, looking at key transcription factors and key chromatin modifiers that can be kind of master regulators of microglial phenotype and microglial primed state that we would certainly be interested in looking at further. So stay tuned. Hopefully we'll have more to share in the future.
Okay, next we have a question from Leticia Weinhardt. Maybe she raised her hand, so maybe she can ask her.
Yeah,
with the
hi,
microphone.
can you hear me?
Yes.
Yes.
Hi,
Yes.
Leticia.
I don't have a great connection, so I hope you can hear me. Now, I was wondering, because you do a lot of, you compare microglia density across regions and across different ages, and there might be difference in terms of structure of the tissue, especially with aging and also different regions. So it might lead to different rate of contraction during perfusion.
Mm.
And that would change the density of microglia, obviously, because you normalize, you look at the number of microglia per area. So I'm wondering if you also try to compare microglia density, but by normalizing with the number of cells, with the total number of cells, instead of looking at number of microglia per area. I don't know if that's good.
Yeah, no, that's this is a really great point that I think we haven't directly addressed. I'm thinking about, we also do a lot of flow cytometry of the midbrain, which contains the VTA and the SNC, and then the whole striatum, which contains the nucleus accumbens. And so we can see these effects of like microglial proportion of cells increasing during aging, specifically in the midbrain. So that's sort of analogous to what you're suggesting with normalizing to like total number of cells, within the tissue by histology. But I think that would be a nice thing to do. We were, I don't think we have specifically tried to assess whether there are different impacts of the aging of the tissue on shrinkage that perfusion would induce. So I thank you for bringing that idea into the pool of things that we should consider.
Because it could be linked to microglia as well, but like I'm sure like the ECM is changing over time, so that would lead to difference of structure. So yeah.
I mean, I think we have we have some other data that make us very confident that the microglia are proliferating. So we've done some BRDU labeling, and then we did have some like lineage tracing where we induce expression of a tag in 2-month-old mice, and then see that the number of those cells and the density of those cells is also increasing. So I'm pretty confident that the cells are proliferating, but I absolutely appreciate the comment that you're making with regard to like the density calculation specifically could be impacted if there are changes in the tissue shrinkage. So that's great. Thank you for bringing that to our attention.
Okay, now the last 2 questions. I have one from Noah Rachmian. Thank you for your interesting talk. Actually, there are 2 questions. She said, I was wondering whether you have performed lipid droplet staining to assess if the aggregates you are observing could be lipid droplet related. And she also asked if, regarding the fire mice, did you evaluate whether the slower ascent to the platform could be due to a motor impairment?
Yes, so for the first question, we have done staining for lipid droplets. Honestly, we were not seeing a large number of lipid droplets during aging, apart from like hippocampus, where a lot of the really beautiful work looking at impact of lipid droplets on microglial aging phenotypes has been carried out. I think we have not dug into this as deeply as one could, but I am viewing them as relatively distinct phenomena. Like we're not seeing any obvious indications that the autofluorescent aggregates contain lipid droplets, or that the microglia that have the big aggregates are more likely to have lipid droplets. We're not seeing any clear evidence of that. But that being said, I have also seen in other tissues, I have seen electron microscopic analyses of these autofluorescent aggregates. They're like electron dense, so you can also see them by ultrastructural approaches. And then if there are little lipid droplets inside the aggregate, you can see that by ultrastructural approaches. That would be something interesting to investigate. And I mentioned that we did proteomic analysis of these aggregates. Probably some portion of the aggregates, possibly even a large portion of the aggregates, is also lipid-based material. We don't really know, not lipid droplets per se, but like lipid-rich material. And that's not something that we currently have information about, how much of these aggregates are protein versus lipid.
Okay, now
and there was another question.
The fire mice.
The second question was
whether a slower ascent to the platform could be due to a motor impairment?
Yeah, we don't think so, just because like on the other behavioral assays, like the open field test, there's not like a difference in the average velocity or like the total distance that the mice run. And certainly just like qualitatively, there doesn't appear to be anything different in the way that they're ambulating. But, there was an earlier question about using more sophisticated approaches to look at gait and motor function, and that's something we could certainly do.
Okay, and the last one is from Isaskun Wendia. You can ask, Isaskun.
Thank you very much for this amazing talk. I have a lot of questions, but I will just post one. It's regarding your aging part of the talk. Do you see any differences between genders, especially between those that have or not have impaired cognitive decline?
Can you say it one more time? My speaker is a little crappy.
Gender. If you saw differences in gender and the aging.
Gender. Yeah.
Gender.
We have included both male and, this is a very important point. We have included both male and female animals in all of our analyses. We're not seeing anything that is just like screaming sex differences, but we're not like optimally powered to really clearly say whether there's like a subtle difference. So I think we can say that there's not like some massive sex differences in the cognitive trajectories. And then similarly with all the like microglial phenotypes that we're looking at, like the lysosomes and the autofluorescent aggregates, nothing massive, but we have not analyzed this yet with enough rigor to be able to say confidently whether there might not be some subtle differences. But yeah, I think it's an incredibly important point, is to understand whether there are sex differences in any of this. Thanks for that question.
Thank you.
Okay, that was the last question. I think, Morgan, you can close the session. Thank you, Lindsay, for the
All right, thank
excellent
you
talk.
so much.
Yes, thank you very much, Lindsay. It was really great. It's really interesting work linking microglia with from development to aging and with different, really different aspects. So I have also many questions, but I think that given that we start late and that you had already many questions, I will just again warmly thank you for your very nice presentation. Thanks everyone for being there today with us, and we will see each other hopefully next month.
Sounds good. Thank you so much for the invitation, and hope to see you all soon.
Okay.
Same here.
Bye,
Thank you
everyone.
very much.
Bye-bye.
Bye.